This is sample for video 1. I obtained this sample from the same pond that I filmed my introductory video. There were many fascinating microorganisms, but the one that stood out most to me were the diatoms. I spoke briefly about diatoms in my sample 2 video 1 post because the predominant form of phytoplankton are diatoms, but I decided these single-celled algae deserved a video to themselves. Let's get into it. Diatoms are characterized by their silica cell walls. They vary in shape and size, but due to their silica cell, are easily recognizable. They create their glass capsules using silicon dioxide and become almost indestructible. Silica is resistant to decay, and because of this unique quality, diatoms have been found in fossils as far back as the Jurassic period. This cell wall is also a reason why diatoms can be found in a vast majority of aquatic environments. They have been found living in ice and are some of the first eukaryotes to be discovered in hot springs. But why are diatoms so important they get their own video? Well, like I said, diatoms are algae and have chlorophyll A and C, so they're able to photosynthesize. They produce oxygen, energy, and most importantly, lipids. Lipids are organic compounds that are insoluble in water. For example, oil, wax, fats, and steroids. Since diatoms produce lipids, they have been used as sources of renewable biofuel. You may also find diatoms in dietary supplements and natural oils. Since diatoms occupy such a large range of ecosystems, they are incredibly diverse. Some live attached to stones and rocks, others act as spores, and those that have a raphe are motile. These morphological differences allow the diatom to thrive in their specific environment. Diatoms are primary producers and account for approximately 20% of global carbon fixation. That's more than all of the world's rainforests. Another interesting quality about diatoms is their involvement in symbiosis with other microorganisms. Larger dinoflagellates and foraminifera may engulf the diatom, or the diatom may engulf smaller nitrogen-fixating bacteria. Being single-celled organisms, diatoms replicate by mitosis. However, since cell replication occurs within the confines of the silica cell wall, each division creates a subsequently smaller daughter cell. To prevent the cells from continuously getting smaller until they die, a suitable mate is used to create an oxospore, in other words, a zygote, and the cell size is restored. Here I've captured a diatom that has moving particles filling the frustule. This could be portions of the decaying diatom, however, I'm unsure of this. If you know what these particles are, leave a comment down below. These incredible microorganisms are essential in numerous research studies, including advancements in nanotechnology, habitat conservation, and medicine. In November of 2015, a research study was published on the use of diatom silica as a delivery system for anti-cancer therapeutics. These diatoms were genetically engineered to code for a binding protein on the silica cell wall that would allow attachment of cell-targeting antibodies. The diatom was a perfect candidate to serve as a carrier, since insoluble organic materials, like lipids and anti-cancer drugs, are perfectly maintained in the frustule. This method was proven highly effective when it was tested on mouse neuroblastoma and lymphoma cells, showing significant regression of growth in tumor cells, all while the mice displayed no symptoms and managed to degrade the silica. This was one of many impacts diatoms had on science, and I'm eager to find out what they could do next. Once again, my resources are in the description below, and stay tuned for my next video where I talk about this guy.